All right, so let's start talking about glycolysis. I like to show this slide to show you how pretty much the rest of the semester is going to look like. We're going to cover a couple things later on in the semester, but we know that glucose is the primary fuel for the cell. Let me say that again. Glucose is the primary fuel for the cell. We all know that. But we're going to begin today to talk about how we get energy out of glucose. So we're going to start with glycolysis. And you can see this happens in the cytosol. So glycolysis happens in the cytosol. And we're going to take a molecule of glucose and make it into 2 molecules of pyruvate. And then that pyruvate is going to go into the mitochondrial matrix. And we're going to then, it gets transformed a little bit, go into the citric acid cycle, also known as the Krebs cycle. And then after that, we are going to go into the electron transport chain to make our goal molecule, ATP. You can see we make measly amounts of ATP from glycolysis in the citric acid cycle, but we make most of it using the electron transport chain. So we're going to do basically part 1 of our metabolism today. And what's different is we're going to learn how we can transfer electrons to these reduced coenzymes. Remember NAD? When we learned about NAD before, and I said that it's holding the electrons in a hydride. We're going to see how that's happening. So we're going to see how we pass electrons from these molecules to NAD or other reduced coenzymes. Basically, they're holding these electrons. We're going to use these electrons in the electron transport chain. That passing of electrons is like a little battery. And that little battery, the energy from the battery is not going to be used to power laser guns as they are used in my house. Super cool laser guns, by the way, we've got for Christmas. Anyway, they're going to be used to power the production of ATP. It's really like a little battery, what's going on in your mitochondria. So what is happening is we are oxidizing glucose down to what molecule? CO2, very good, because CO2 is the ultimately oxidized carbon. Carbon with 2 oxygens, right? It's 2 double bonds, carbon with 2 double bonds to oxygen. So that's an ultimately oxidized carbon. So we're going to see how we're taking glucose and oxidizing it. While that oxidation is happening, something else has to be reduced, right? You can't have oxidation without reduction. So the things that are going to be reduced are the coenzymes. So the first pathway in glucose metabolism is glycolysis. And a pathway is just a series of reactions. Again, the next one is going to be the citric acid cycle. We're going to use the word citric acid cycle rather than Krebs cycle. And this glycolysis happens in 2 stages. One where we have I'm hoping this works. When we split the molecule into we have glucose, the first stage of glucose is split, and then the second stage we get 2 molecules of pyruvate. So again, this is the main way organisms extract energy. So let's see if this works. If this link isn't working, I can find another one for Friday. I thought it was working. Again, I checked at the beginning of the semester, but I didn't check this morning. And our super fast. It looks like it's working. So what this YouTube is going to do is walk us through glycolysis. And then we're going to go through it many times before. But I'd like to show an overall view of it. And then we can, oh, that's an advertisement. I love that these scientific
No absence feel like Glade.
What can I skip?
Melt your mood with a Hawaiian breeze fragrance.
It's not going to let me skip it.
Feel relaxed. Feel Glade.
We're all going to buy Glade now.
Cells break down food molecules,
So
such
this
as
is glucose,
through multi-step pathways. In the process of glycolysis, the breakdown of one glucose molecule into two 3-carbon molecules produces a net gain of energy that is captured by the molecules ATP and NADH. In eukaryotes, the breakdown product, pyruvate, is imported into mitochondria, where it ultimately feeds into the citric acid cycle and the electron transport chain. Glycolysis involves a sequence of 10 steps. In the first 3 steps, energy in the form of ATP is invested to be recouped later.
So we're
In the
putting money
4th
in.
and 5th steps, this energy allows glucose to be split into 2 smaller molecules from which energy can be harnessed efficiently. And in the last 5 steps, energy is released stepwise as ATP and NADH.
We're getting our money back.
The elegant chemistry that evolved to catalyze these reactions ensures that energy is released in small portions that can be efficiently captured. Less controlled combustion reactions would release most of the energy as heat.
So if we just burn glucose.
In the first step, the enzyme hexokinase uses ATP to phosphorylate glucose.
Our dangly
This investment
carbon.
of energy primes glucose for energy-releasing reactions later in glycolysis. The resulting molecule is glucose 6-phosphate.
So this dangly carbon is phosphorylated. Number 6.
ADP is released. This first step of glycolysis is irreversible. In the second step of glycolysis, the enzyme phosphoglucose isomerase catalyzes the opening of the ring form of glucose 6-phosphate to the open chain form. The same enzyme then performs a reversible reaction in which the carbonyl group of glucose 6-phosphate changes position from the first carbon to the second carbon in the chain. This reaction involves a water molecule, which donates a hydrogen ion to the carbonyl oxygen.
I'm not going to require you to...
The hydrogen ion is then retrieved from the hydroxyl group on the second carbon, creating a new water molecule.
We're just
In the
moving
process,
the carbonyl down.
fructose 6-phosphate is formed. The same enzyme, phosphoglucose isomerase, catalyzes the formation of fructose 6-phosphate into its ring form.
So we go from glucose to fructose. Another dangly carbon. What's going to happen?
In the third step of glycolysis, the enzyme phosphofructokinase
See that?
uses ATP to phosphorylate fructose 6-phosphate. ADP is released, and the molecule fructose 1,6-bisphosphate is formed.
Now look, it's
Third step,
symmetrical,
in
and
which
the
the
dangly
second
carbons
phosphorylation
are
event occurs,
phosphorylated.
is irreversible and is a major regulatory point in the commitment to glycolysis.
Now we're going to
The
chop it.
phosphorylations in steps 1 and 3 represent an investment of energy that will be paid back in the later stages of the pathway. Step 4 of glycolysis begins with the opening of the ring form of fructose 1,6-bisphosphate into its open chain form. In this step, the enzyme aldolase cleaves fructose 1,6-bisphosphate into 2 molecules.
Let's just-
One molecule that is formed is the 3-carbon glyceraldehyde 3-phosphate. The enzyme performs additional reactions on the second 3-carbon molecule.
Very cool. So we
The
now
second
have 2 molecules.
is dihydroxyacetone phosphate. In step 5 of glycolysis, the enzyme triose phosphate isomerase catalyzes the isomerization of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate.
Because we don't want to throw that other one away.
The catalytic mechanism of this enzyme is very similar to that of phosphoglucose isomerase back in step 2. The result is 2 molecules of glyceraldehyde 3-phosphate.
Now we're ready to go into stage
All
2.
of the subsequent steps of glycolysis will occur twice, once for each molecule of glyceraldehyde 3-phosphate. These are the energy generation steps of glycolysis. In step 6, the enzyme glyceraldehyde 3-phosphate dehydrogenase uses NAD to oxidize glyceraldehyde 3-phosphate. The resulting molecule is connected to the enzyme by a high-energy thioester bond. A molecule of inorganic phosphate displaces the high-energy thioester bond, forming a high-energy acyl anhydride bond.
It's very strange.
The resulting molecule is 1,3-bisphosphoglycerate.
Look at what all that's going on in that
In the
molecule.
seventh step, the enzyme phosphoglycerate kinase dephosphorylates 1,3-bisphosphoglycerate. The high-energy phosphate is transferred to ADP, forming ATP.
Money. That's our money molecule.
The 3-carbon molecule is now 3-phosphoglycerate. Because this reaction occurs twice, once for each 3-carbon molecule, a total of 2 ATPs are generated. At this point, the energy investment from the first 3 steps has been paid back.
So now we're even.
In the 8th step, 3-phosphoglycerate, which has a relatively low free energy of hydrolysis, is transformed by the enzyme phosphoglycerate mutase into 2-phosphoglycerate.
So we're moving the phosphate up.
In the 9th step, the enzyme enolase removes a water molecule from 2-phosphoglycerate, creating phosphoenol pyruvate. The loss of water redistributes energy within the molecule, creating a phosphate group with an extremely high free energy of hydrolysis.
It's very strained.
In the 10th and last step of glycolysis, the enzyme pyruvate kinase transfers the high-energy phosphate group to ADP, forming ATP and pyruvate.
Now it's more relaxed because it gave the phosphate.
In the second half of glycolysis, many of the reactions release energy, captured in the form of ATP and NADH. Overall, the net energy produced in glycolysis from a single molecule of glucose is 2 molecules of ATP and 2 molecules of NADH.
Not much.
The chemistry of glycolysis is conserved all the way from bacteria to animal cells.
Okay, I like to show that, not because I want you to memorize everything that woman said, but because it gives you a nice overview of what's going to happen. I'd like you to see the whole thing overall, and then we'll go back and take a look at each step. So what we're gonna do now is go through in words what's happening, and then we're gonna go through and show schematically. So in the first step, we're phosphorylating glucose. Remember, you saw the carbon number 6, it's dangling out there, right? So we're dangling out there that carbon number 6, that gets phosphorylated. Now, one of the things that happens with that is that once glucose enters the cell, it gets phosphorylated and it keeps it inside the cell. So this first phosphorylation of that carbon number 6 to make glucose 6-phosphate keeps the glucose in the cell. But we're spending money, all right? Remember, our money molecule is ATP. So we're inputting money. It's just like you guys going to college. You spend money to make money, right? You're spending money on tuition to hopefully make a lot more money later on, right? Let's hope. Let's hope that we're doing that. So look at the stage 1. It's sort of like your tuition, where you're spending some money. We're going to spend, this is our first spending of money. Maybe this is Hudson Valley, and you're going to go on to a 4-year school afterwards, right? So our first spending of money. Now, this isomerization, we're going to get our fructose, because fructose is a ketose. It makes another dangly carbon. So a lot of the reactions in our pathways, I call it getting ready reactions. And this one is one of the getting ready reactions, because what we're doing is we're making another dangly carbon, which then gets phosphorylated again. So this is our second input of ATP. So we phosphorylate, we isomerize to make another dangly carbon, and then we phosphorylate that dangly carbon. Do you like my technical terms? Dangling. It works for you. It's got to be dangling out there in order to get phosphorylated. It can't be buried in the ring. It's hard for the ATP to get there. So if you see something dangling out there, think that's going to do something, right? All right, so once we have fructose 1,6-bis, bis means 2. The 1,6 tells us where it's happening, on carbon number 1 and carbon number 6. So fructose 1,6-bis phosphate is then chopped in half. So this cleavage is chopping in half. Unfortunately, we don't chop into 2 equal halves. Glyceraldehyde 3-phosphate is our entry molecule for stage 2. But when we chop in half, we get glyceraldehyde 3-phosphate and another molecule, dihydroxyacetone phosphate. We're not going to throw things away. The cell is very efficient, all right? Minimal waste, no side products, right? So what we're going to do is, in the next step, is we're going to take that dihydroxyacetone phosphate and make it into another molecule of glyceraldehyde 3-phosphate. Then, in the next step, we're going to add a phosphate as it is oxidized. So we're going to add what we call an inorganic phosphate. Now, one of the things I need you to kind of bear with me is that when in these reactions, hydrogen ions kind of come and kind of go. We don't account for them. We don't balance them because we are in an aqueous environment that has protons. Also, do you remember what the buffer in the cell is? It's a phosphate buffer. So phosphates also come and go. We don't account for them coming in, and we don't account for them going out. So we are not necessarily balancing reactions as we did in Chem 1. So here we get an inorganic phosphate. What do I mean by inorganic phosphate? Just a PO4, maybe with a couple hydrogens on it, going around in solution, all right? I write P with a little I next to it, and you'll see that as my shorthand, and you'll see that when I'm writing it around. When it's not an inorganic phosphate, it's attached to, say, ATP. That's not an inorganic phosphate. That's an ATP phosphate. So what we're doing in this one is we're getting this additional, to make bisphosphate, phosphoglycerate, we're getting that just from the solution. So this is happening in duplicate because we have 2 glyceraldehyde 3-phosphates. Then that inorganic phosphate, because it's on what I call an angry molecule, and I'll talk about that later, it's very strained. It has something called a high energy of phosphate transfer potential. And we'll talk about that a little later, but for now we'll call them angry. They're strained. This one, the 1,3-bisphosphoglycerate, is going to give up one of the phosphates so that it's not so strained. And it's going to give it to ADP to make our money molecule. It's making an ATP. But remember, it's happening twice because it's in stage 2. Then we have a getting ready reaction. There's actually a couple getting ready reactions. We're going to move the phosphate from the 3rd position, the 3rd carbon, to the 2nd carbon. And then we're going to remove a water molecule. And then it makes a super angry molecule. Phosphoenolpyruvate, I call it PEP, is got a lot of free energy in it, a lot of strain. And for that, it's going to lose that phosphate. And it's going to give that phosphate to ADP to make ATP. So now we've made our 3rd and 4th molecule of ATP. That gives a molecule of pyruvate for each one time it happens. This is how, as we go through our reactions, this is how I'd like you to remember them, okay? By what's happening and why. What's happening and why. So here's another look at what we just went through. We're going to take a molecule of glucose and then put an ATP to make glucose 6-phosphate. One thing I want you to notice is that in a couple spots there's only one arrow. The other ones have double arrows. If it's only got a single arrow, it means it's irreversible. And things that are irreversible means that the products have much less energy than the reactants. So because of that, we can't go backwards. Remember, a reversible reaction is reversible because the energy of the products and the reactants are about the same. So we can go back and forth. But when you have products that have much less energy than the reactants, you can't roll the ball back up the hill unless you put in energy. So when you see the single arrow, know that they're irreversible. Now, as we go further in our study of glycolysis, we're going to find out that these points that are irreversible are also controlled by enzymes that are under allosteric control. So these single arrow points are going to be most likely where we are going to control. So this phosphorylation is under control. We're going to look at that later. Also, points where you're investing money, investing ATP, those are going to be the points of control as well. So you can see this one, we're investing ATP. We have products that have lower energy. We are keeping glucose in the cell. Then we're going to isomerize glucose to fructose to create another dangly carbon. We're going to input another phosphate to make fructose 1,6-bisphosphate. Then we're going to cleave. We're going to chop it in half. Half of it's ready to go into stage 2. The other half is not. This is not our entry molecule. So you can see in step 5, we're going to isomerize this dihydroxyacetone to our entry molecule, glyceraldehyde 3-phosphate. Now you can see what we're going to do in step 6 is to add that inorganic phosphate. But what also is happening is as we are adding that inorganic phosphate, we're oxidizing. Something else has to be reduced. And so we call this an oxidation and phosphorylation. So we're phosphorylating, so we have 2 phosphates, but we are also oxidizing. When we oxidize, we have to reduce. So here's our NADH. Right? This is going to be a money molecule. Not as money as ATP, but we're going to then turn it in for money. So maybe it's Canadian quarter that you can't use. It's a Canadian quarter, so we have to turn it in later. This is our American dollar. All right, this is a really good one. All right. Then we get some money back. All right. This one, glycerate 1,3-bisphosphate, it has a high phosphate transfer potential. In other words, it's angry. All right, a lot of energy. Then we are going to isomerize to make another angry molecule, which is PEP. So step 8 and step 9 are reactions that produce another angry molecule. So we isomerize to put the phosphate on the second carbon, and then we dehydrate to put a lot of strain in this molecule. So once we create another angry molecule, high phosphate group transfer potential, we are then going to take that last phosphate off and give it to ATP. So you can see we've put in 2 ATPs, got now 4. We've also got now 2 NADHs. So putting in 2, getting out 4 is a net of 2 ATPs. We also get a net of 2 NADHs, and then these pyruvates are going to go on in the next stage into our citric acid cycle. Now I bet you this is about as far as you've gone with your other courses for glycolysis. What we're going to do next is probably take it a step beyond what you've seen before. Did you guys take a look at glycolysis in this detail for your other classes? So what we're going to do is go on and look at exactly what's happening. We're going to focus on the energetics and the control through our enzymes. So if you guys that have not gotten a handout, so if you can bear with me, what we're going to do is take a look at each one. All right, so in step 1, again, we're taking this dangly carbon, and I'll show you how to fill out the paper, and you'll have plenty of time to do that. We're going to take this dangly carbon, carbon number 6, and we're going to phosphorylate it. it requires an ATP. Now, just taking and putting a phosphate onto carbon number 6 is what we call endergonic, meaning it's a positive delta G. So this word endergonic means positive delta G, means non-spontaneous. Endergonic, positive delta G, not spontaneous. Requires energy. What we are doing is we are coupling it to another reaction, which is the hydrolysis of a phosphate from ATP. So this idea where you take and drive a reaction that's not spontaneous and you couple it to a reaction that is spontaneous is something that is you're going to see throughout all the pathways, okay, throughout all the pathways that we are going to study. So we are coupling an endergonic reaction of phosphorylation to an exergonic or negative delta G. So you can see maybe the positive delta G is this big, right? One hand width. But the negative delta G is 2 hands. So that when you add it together, you get a negative delta G. Right? So we have, let me write that down. I don't have the energetics. I know that hydrolyzing an ATP gives you out 30.5 kilojoules per mole. All right? But say it takes a positive 20, I don't know what it is, I'm making that up. Say it takes a positive 20 kilojoules to phosphorylate at that. If you add those together, it's still a negative 10. So that's the idea of coupling two reactions. And so that the overall reaction is still exergonic or negative delta G, meaning spontaneous. Now this is sufficiently exergonic that the arrow goes one way. Meaning that the products are lower in energy than the reactants. Remember, every molecule has an internal amount of free energy, delta G, of formation. Remember that from general chemistry? And so these two molecules have a sufficiently lower, put together, sufficiently lower free energy than these to create a situation where this reaction is irreversible.
Can you say that again?
That these two molecules, our glucose 6-phosphate and our ADP, are sufficiently lower in energy, in delta G of formation, than our reactants, so that the reaction is irreversible. So our products are lower in energy than our reactants. And because they are, the reaction is irreversible. It goes forward only. It's exergonic. Overall, it's exergonic. Negative. When I say exergonic, out, negative free energy. So there's a couple definitions. Endergonic means positive delta G. Exergonic means negative delta G. Endergonic means non-spontaneous, positive delta G. Exergonic means negative delta G and spontaneous. So what we're doing is taking this endergonic reaction, coupling it with a more exergonic reaction, more negative delta G, to create an overall negative delta G reaction. We'll take a look at the overall negative delta G later on. Another thing I want to talk about is this hexokinase. We're going to talk about hexokinase a little bit later, but this is our first enzyme that you're going to need to memorize. There's only 3 enzymes out of this whole pathway that I need you to remember, and this is the first one because it's a point of control. So as we're studying the specifics of our pathways, there's 2 things I need you to keep in the back of your mind. The free energy and the control. So we're going to know that this one is negative delta G, and that this is a point of control. Hexokinase is subject to allosteric control. So what we're going to do now is go over to our document camera. What I want you to do, so what I have here is I want you to draw glucose and glucose 6-phosphate. You can look at your notes too to help you out. And the enzyme hexokinase is going to go into the oval, and the dotted one is going to say what the reaction step is. And then here, this is ATP is going in and ADP is going out. We don't have a lot of time to write these things down, so if you are having trouble getting everything written down before we move on, you can either go back to the slides to write things down, or you can take a picture of my papers. Again, I'm not going to ask you to spit this back out. And in fact, on the exam, rather than writing structures, I'm going to focus more on the names rather than the entire structures. Where I say inhibitor.
And the second structure attached to the sixth one, Steve.
Oh, yeah, it's a phosphate group. So it's oxygen, phosphate, double bond to oxygen, another 2 oxygens. I don't have the charges on there either, so just know that it's a phosphate group on there. Yes, Alex?
What did it say next to the inhibitor?
Yeah, the inhibitor is glucose 6-phosphate and ATP. So those are inhibitors of this. So I haven't talked about that yet. So next to where it says inhibitor, glucose 6-phosphate and ATP. So because this enzyme is under allosteric control, it's going to have an inhibitor. Some have both activators and inhibitors. This one only lists its inhibitors. I just want to talk a little bit about that. It makes sense that ATP would be an inhibitor, because if we have a lot of ATP in our cell, why are we going to use glucose to make more ATP? Yes, Aaron?
Does it say G6?
Yeah, glucose 6-phosphate. We are inherently lazy to write down the full word, glucose. So what I want you to remember is a phosphorylation, phosphorylating carbon number 6. Hexokinase is the enzyme for control. We are going to have to later on know the inhibitors, and that is an irreversible reaction, meaning that it gives off energy, and we're investing ATP. All right, I'm going to go back to so this second step, we're converting an aldose to a ketose. An aldose to a ketose, meaning that we're going to have the anomeric carbon on carbon number 2. And it's done doing a 2-step process. I don't need you guys to know that. Right? I don't need to know, you didn't know this. But basically what happens is that this ring structure is going to go to the open chain. And then the open chain is going to move the carbonyl from the first carbon to the second carbon. And then we're going to close it. Notice that we have a 2 arrow. That means that the energy of this one, of the product and the reactant, are about the same. What we're doing is we're reducing and then oxidizing. So it's just an isomerization, where we take away the carbonyl from the first one, and then we add the carbonyl to the second one. What I want you to remember is that this is a reversible reaction, We're going from glucose to fructose to make another dangly carbon. And that's an isomerization. What's going to happen next, guys? Phosphorylation. So see, you guys are learning it. Why? Why? But before we do that, we're going to go back to our document camera. And do that isomerization. I don't have any inhibitors here because this enzyme does work to catalyze this reaction, reduces activation energy. However, it is governed by Le Chatelier's principle, whether it goes forward or reverse. So when we see the double arrow, it's going to be Le Chatelier's principle, meaning that if you have more reactants, it's going to go towards products. I like to see these pathways when I talk about the free energy. The free energy is like a river or a stream going down a mountain. Those reactions that give off a lot of energy, those irreversible reactions, are like a little waterfall, right? And those reactions that are governed by Le Chatelier's principle, the reversible reactions, are the ones that are going through the flat land. It's still going to go down the hill, right? You can have a stream going flat, but it will still continue because it either has a waterfall behind it or it's a waterfall ahead of it.
So you said there's more products than reactions?
If there's, this last reaction is governed by Le Chatelier's principle. So if there's, compared to the equilibrium constant, if there is more reactants than we should have, then it's going to go forward. It all depends on the equilibrium constant, which I don't know. All right, in the next step, we're going to phosphorylate this dangly carbon. What do you notice? Single arrow, right. It's one way, irreversible, because we're investing another molecule of ATP. We're going to phosphorylate, just like you guys guessed, that dangly carbon. And I'm going to call this enzyme PFK because if I don't, I end up saying a bad word. So I call it just PFK because I can't pronounce it. So this is going to be under allosteric control. We're going to study that in a bit. So we're going to go back. We call this fructose 1,6 bisphosphate. The bis stands for 2. Actually, before we go forward and write this down, I'm going to go through a couple more slides. Let's talk about PFK. It is a key enzyme, just like hexokinase. It has positive and negative feedback, but I just want to point out, you don't necessarily have to remember this, but it has 4 subunits, and they're designated either L or M. There's not a lot of difference between them, but we have PFK that exists in all of the M subunit or all of the L subunit. And remember, I may have talked about how in my graduate study I talked about tubulin and how all the work had been done with tubulin from the brain, but then I realized that the amino acid sequence was slightly different depending upon where the tubulin was obtained from. That when I isolated it from cancer cells, I realized that the binding of the anti-cancer drugs was a little bit different to the tubulin from the cancer cell than from the brain. So cow brains, not people brains. And so this is the same sort of situation where the amino acid sequence is slightly different based upon where you're going to get it from. It can either have all 4 subunits be this M amino acid sequence or all 4 subunits being all the L. And they're called isozymes. And when I looked at tubulin, it's not an enzyme, so I call it an isotype. So you can, depending on where you find it, it's going to be made up a little bit different. It functions the same. It catalyzes this reaction, but it's made up a little bit different. M, you find this one in muscle, and the L in liver. So ATP affects it. If you have a high amount of it, it's going to inhibit. And fructose 1,6-bisphosphate is also an inhibitor. Actually, this is a typo. This should be 2. Fructose 2,6-bisphosphate is an activator. And I'll talk about that in a little bit. Actually, no, I'll talk about it now. Fructose 2,6-bisphosphate is produced by hormones when glucose in the blood is high. Okay? So fructose 2,6-bisphosphate is produced by hormones when glucose is high. And actually, in red blood cells, you'll find different combinations of the isozymes. So here's the different types. So here's the heterogeneous forms. You have 3M, 1L, different combinations of the two. But here, this is what you find in muscle, and this is what you find in liver. So these are called isozymes. Does anybody have a question? No? All right. So far, and I have it right here, that we have 2 of the 3 enzymes. We've talked about 2 of the 3 enzymes that are under allosteric control, are important for you to learn in our glycolysis. So the first one was for the first reaction, hexokinase, so that we are going to control the first use of our ATP. We know that ATP is going to be an inhibitor. Why invest ATP if we have a lot around? We don't need to do that. Also, if we have a lot of our product around, we're going to inhibit. So a lot of these you'll see that either its product or maybe a downstream product will be an inhibitor. If I asked you, what's an inhibitor of this enzyme in this metabolism reaction? And if you have no idea, what molecule are you going to write? ATP. Because why have metabolism at all to make ATP if ATP is around? So pretty much for almost all of our enzymes that are under control, ATP is going to be an inhibitor. I would like you to know the other ones too. But you can see that glucose 6-phosphate is the product for the reaction that hexokinase catalyzes. So if we have a lot of that around, it's going to inhibit that. Now, PFK is the one for the third reaction. And I don't want you guys to remember these reactions by numbers, okay? That can't be the way you answer the questions. But I want you to say, okay, so it's going to be the fructose 6-phosphate to fructose 1,6-bisphosphate. That's how I want you to remember these. Now, ATP, of course, is going to be an inhibitor, but also citrate is an inhibitor. Citrate is an intermediate in the citric acid cycle. So if we have a lot of citrate around from the citric acid cycle, we don't need glycolysis to happen. We said that fructose 2,6-bisphosphate is produced when glucose in the blood is high. So we want to, it's almost like a feed forward. We're saying, oh, we need to use this glucose up to produce energy. We need glycolysis to happen because we have a lot of glucose in our blood. So after you have a candy bar, that's going to be produced to say glucose, and then my kids will jump around the house because they have a lot of ATP. So that's where you get that sugar rush of energy. AMP is adenosine monophosphate. It's our mono molecule without the phosphates on them. So we don't. It's energy deficient molecule, right? So it doesn't have those phosphates on it. We'll talk about pyruvate kinase later. So those are those 3 reactions. Okay, so let's go back to our document camera. Okay. So here we're making, oh goodness, my handwriting is terrible. This is my 1,6-bisphosphate of the fructose. That's my PFK. If you guys just want to write down PFK, memorize PFK as the enzyme, that's fine. And it's a phosphorylation ATP comes in, ADP goes out, so we're investing energy. And the activators are the fructose 2,6-bisphosphate, AMP. The inhibitors are citrate and ATP. Activators, fructose 2,6-bisphosphate, AMP, inhibitors, citrate and ATP. This next step is where we're going to take, it looks pretty symmetrical, doesn't it? The fructose 1,6-bisphosphate, you got a phosphate on the 6, a phosphate on the 1, oxygen in the middle. We're going to take a little cleaver and chop it down the middle. And so we're going to make 2 3-carbon molecules. This glyceraldehyde 3-phosphate is going to be our entry molecule for the second stage. So we're going to chop it down, 1, 2, 3, 4, 5, 6. So the first molecule, our dihydroxyacetone, is made from the first 3 carbons, and then our glyceraldehyde 3-phosphate, that's going to make our glyceraldehyde 3-phosphate. And again, this is a reversible reaction, and I'm not going to talk about the mechanism. So remember, this one is cleavage. You're going to chop it and make 2 uneven molecules. What we're doing in the next step is we are going to take that dihydroxyacetone and isomerize it. We're going to move this carbonyl up to carbon number 1 and the hydroxy down to number 2. Before we go back to the document camera, I want to mention, all right, so now we have 2 trioses. So this is going to go into the second stage. So this reaction, they say, has a small positive delta G. That means it's slightly non-spontaneous. But remember that if you imagine your stream coming down a mountain, even if there was a slight uphill, if there's a waterfall ahead of it and a waterfall behind it, even if there was a slight uphill, it would still go forward, right? And this is what's happening here. There's a slight uphill going from the reactant to product, but because downstream and upstream there's waterfalls going down, a lot of free energy release, that this one still goes. Now, another point I want to make about these delta G values is that they're sort of artificial. A lot of them will have this little circle right next to the delta G. What does that mean? If I have a little circle right next to the G, it means not initial. It means standard conditions. What are standard conditions? Do you remember? 1 atmosphere for gases, 1 molar concentrations, right?
25 degrees Celsius.
And 25 degrees, which we're a little bit warmer than that, right? And we are also not 1 molar because we'd be dead. So these delta G values are slightly artificial because they are at different conditions that are in the cell. So you're going to take these delta G values with a grain of salt, all right? They give us a clue. But here we've taken our glucose and we've split it into 2, in 5 reactions, split it into 2 molecules of glyceraldehyde 3-phosphate. We've had to input 2 molecules of ATP. And then we have hexokinase and PFK that are subject to allosteric control. The PFK is very crucial to this. All right, let's go back to our document camera and finish up stage 1. Basically, what's happening here is this is our fructose 1,6-bisphosphate. We're making the dihydroxyacetone phosphate that we have to further isomerize to glyceraldehyde 3-phosphate. And then we've also cleaved it to our glyceraldehyde 3-phosphate. So these are the 2 molecules that we're getting from cleaving this. That's why I have an arrow going down here and an arrow going down here. Adalase is the enzyme for this reaction, but here and it's a cleavage for this reaction. But here we have to have another reaction going down here. There's one thing that I'm missing, and it's the double arrows. There should only be 2 single arrows on this page. Yes, Aaron?
So double arrows go on the tri.
For this one and this one and this one. They're all, this is sort of hard for me to write. It's really a double arrow down to make 2 products. So it's just, it's one reaction making the 2 of these. I just didn't know how to write that. I'm not that creative or skilled at making things. So this is, it's 2 double arrows. It's one, it's a double arrow going down here and here making these products. But this, making this from this is reversible. And also making this from this is reversible. But where we've invested the ATP, those are only going to be single arrows.